Hello everyone and welcome to the Student Physio. Today's topic is experimental physiology. This has been my personal favorite topic as this has maximum weightage for your spottings. Out of 10, 4 to 5 spots are based on these graphs. So what is experimental physiology? It uses amphibians, that is frogs, to demonstrate properties of the cardiac and skeletal muscles. Now what are these properties? These properties are excitability, contractility, conductivity, autorhythmicity, and so on. We'll look forward to this. Now you might question why frogs were used. Because frogs were poikilothermic, that is, these are cold-blooded animals. They can withstand the changes in temperature and pressure very well. Also, they on the cellular level, they are physiologically very similar to human beings. Next, these are easily available, they were cheap, they were harmless and easy to handle. And now the type of stimulus that is used to record the changes can be electrical, mechanical, chemical, or thermal. So in this, electrical stimulus is the major types of stimulus used. Now why is that so? Because imagine if you have to stimulate anything mechanically, like me compressing a ball. I don't know what the force, what the strength of energy I am using to stimulate the ball. I cannot even quantify it. I cannot measure the strength, and I cannot ensure that these same strength can be applied again and again by me. It can vary with the amount of fatigue in my muscle. So electrical stimulus is therefore very much preferred because the duration, strength, and frequency, it can be controlled with an electrical stimulus. It can be repeated with the same strength and frequency. Damage to the tissues is a minimum with the electrical stimulus, as where if we use chemical stimulus like acids or any other bases, the risk of injury to the tissues is high, which is overcome, or which is very minimal in case of the electrical stimulus. That is another reason. One more is it can be applied to a limited area. Like we talked about mechanical or thermal stimulus, if we say, the amount of area that I'm putting the solution, or the amount of area that I am heating cannot be controlled. But with a mechanical stimulus, but you can do it precisely by using electrical stimulus, by using small electrodes. So now let me now make you understand how is the heart prepared. So now for preparing the heart to obtain the graphs, the frog is first pitched. What is a pitched frog? A pitched frog is basically a brain dead frog whose so reflexes and other actions are dead, but some functions like the heart stays intact for some time. Once the frog is pitched, the chest cavity of the frog is cut, and the heart is exposed. The exposed heart, at the tip of the ventricles, a U-pin is inserted. With the U-pin, the heart ka surface is lifted up, and the electrode is placed on the tip of the ventricle with the help of the U-pin, and the heart is raised above from the surface. Then this U-pin is attached to a very thin thread. This thin thread is further attached to a marker pen or a recording instrument, which can further record the contraction and relaxation of the cardiac muscles. The recording instrument is a drum. Moving drum is used to record the systole and diastole of the heart. Like when the heart contracts, imagine this is the heart. This is an imaginary thread which is attached to a pen and a recording drum. When my heart contracts, the thread is being pulled, the pen pulls, and there is a stroke made on the drum or on the sheet of paper which is attached to the drum. So as my heart contracts, the thread is pulled, there is a stroke. As it relaxes, the thread is released and there is a stroke again. This makes the normal cardiogram. The normal cardiogram is a picture of the normal systole and diastole of a beating heart, where the contractile functions of the heart stays intact in a pitched frog. So when the heart contracts, there is a systole and an upward stroke is made on the normal cardiogram. Then when the heart relaxes, there is a downward stroke. A special point to be noted is that like in human heart, the pacemaker of the heart is SA node. Similarly, in frogs, the pacemaker is said to be the sinus venosus. Though the frogs have a three-chambered heart and it doesn't have a specialized conductive system like humans do, like the AV node, bundle of hairs, and the Purkinje fibers. It is not present in heart of frog. The frog's conductive system is simple: sinus venosus, then the atrial musculature, and then the ventricular musculature. As the sinus venosus sends the impulses, the atrial musculature contracts, then the ventricular musculature contracts, and there is this systole noted on the graph. And as it relaxes, there is this diastole noted. This is the normal cardiogram. Points to be remembered in this normal cardiogram is: the upward stroke marks the systole, downward stroke marks the diastole or relaxation. A change in the speed of the recording drum. If the speed of the recording drum changes, like if the drum is moving more faster, the length of the record that is recorded it spreads. The systole continues for a longer period of time, and then the diastole continues for a longer period of time. This helps to study the individual strokes very clearly, while the decrease in the speed, this is the decrease in the speed of the drum, it crowds the strokes more. Therefore the records are more crowded and you can barely see anything clearly, like the contractions and relaxations both appear to be the same. Also, the difference that has to be noted between the electrocardiogram and cardiogram. What is a cardiogram? A cardiogram is basically the record of mechanical activity of the heart, while the electrocardiogram is the record of electrical activity of the heart. Another points that will be important further are the height. This height determines the force of contraction. If the height of the systole recorded is decreased, this can be noted as decrease in the force of contraction, while the number of the peaks and the systole and diastole noted on the graph, if that is increased, it can be noted as the increase in the heart rate. The number symbolizes the heart rate, while the height symbolizes the force of contraction. So when we check the effect of the effect of warm and cold ringer solutions on the normal cardiogram, there will be changes seen in the height and the number of stimulation or number of systole and diastole present on the graph, which will be then determined as the change in the heart rate or change in the force of contraction. As we now have finished with the cardiac muscle, how and has seen how the stimulations are recorded with the cardiac muscle, now we have to see about the skeletal muscles. The skeletal muscle now used here is the gastrocnemius, which is the calf muscle of the frog, and the nerve used is the sciatic nerve. The sciatic nerve and the gastrocnemius preparation is as shown in the picture. The connection of the sciatic nerve to the gastrocnemius is kept intact, and when the nerve is stimulated either at the vertebral end or the muscle end, it produces a contraction of the gastrocnemius, which is further recorded, which is then called as a simple muscle curve. Let us now see about a simple muscle curve. How a simple muscle curve looks on the graph. This is the most common graph to be asked in the spottings. While the point A is said to be point of stimulation. That is when the electrode or the electrical stimulus is applied to the muscle. Point B is referred to as point of contraction. That is the beginning of the shortening of the muscle. Point C can be referred to as the peak of contraction, that is the maximum contraction point, or the starting of relaxation, or just the contraction. And point D can be referred to as the end of relaxation phase, and these further waves are called the shatter waves. So the period between A to B, that is the point of stimulus to the point of contraction or beginning of contraction, is referred to as latent period. So let me now make you understand the concept of latent period very nicely. लेटेंट पीरियड इज व्हाट द डिले कॉस्ट व्हेन द मसल इज स्टिमुलेटेड. मैंने अभी स्टिमुलेट किया और उसको जो कॉन्ट्रैक्ट एक्चुअल कॉन्ट्रैक्शन करने में जो टाइम लगा उसके बीच का टाइम, दैट इज द लेटेंट पीरियड. नाउ व्हेन आई स्टिमुलेट द मसल, मतलब मैंने नर्व के एंड पे नर्व को स्टिमुलेट जब किया है, पहला तो नर्व को खुद को टाइम लगा to take that impulse. That is the first. That is the true latent period of the nerve. Is the first cause for the latent period. Second, when this impulse is taken at the one end, to transfer or transmit that impulse from one end to the end of the muscle or the muscle end of the nerve, इस ट्रांसमिशन में जो टाइम लगा, दैट इज द सेकेंड पॉइंट ऑफ डिले. देन थर्डली, अगर अब इस एंड पे पहुंच भी गई है इम्पल्स, दिस इम्पल्स टू ट्रांसमिट इनटू द मसल एट द मायोन्यूरल जंक्शन. मायो दैट इज मसल एंड न्यूरल का जंक्शन, नर्व और मसल के बीच का जंक्शन, to transmit this impulse from the nerve to the muscle, this is the complex procedure, and इसमें जो time लगा, that is the third point of delay. Therefore the 3 points being: first, the true latent period of the muscle; second, the transmission time or conduction time of the nerve; third, delay at the myoneural junction; fourthly, अभी मसल में कॉन्ट्रैक्शन और या जो एक्साइटेशन करने वाला ये था, दैट इम्पल्स इज रीच्ड. Now the time from that excitation point to the actual contraction or shortening of the muscle, or the cross bridge formation and relaxation and actual shortening of the muscle में जो time लगा, that is again the fourth point for delay. So the fourth point being the excitation contraction coupling का delay. Now what is excitation contraction coupling? It is basically when the muscle is excited or the nerve impulses reaches the muscle, it has to contract, and this coupling is called the excitation contraction coupling. The fifth point of delay being the inertia of the recording instrument. अब सब कुछ हो गया, नर्व को स्टिमुलस भी मिल गया, उसने इधर ट्रांसमिट भी कर दिया, उसने मसल को भी पहुंचा दिया, मसल को स्टिमुलस मिलने के बाद उसने कॉन्ट्रैक्ट भी कर दिया, बट नाउ दिस कॉन्ट्रैक्शन हैज़ टू बी रिकॉर्डेड. दिस recording is done by a mechanical instrument where we are using this thread or pen and then a recording drum. So this inertia of the instrument to record, इसमें जो time लगा, that is also contributing to the total latent period caused in this muscle twitch. After the latent period, the further from the point B to point C, this upward steep graph is called the contraction period. This is the period when the actual contraction of the muscle is happening, or that is the shortening of the sarcomere is happening. Then from point C to point D is the relaxation period. It is the time taken to relax the muscle, where there is removal of the calcium from the sarcoplasma and the sarcoplasmic reticulum, which is an active process. That is, it also needs ATP or energy, and the factor that is responsible for relaxation is the load acting on the muscle. This is the simple muscle twitch that is produced. So today we have discussed about the normal cardiogram and the simple muscle twitch, which are used to record both the cardiac muscle properties and the skeletal muscle properties. So we have 11 graphs for the cardiac muscle and to study the properties of cardiac muscle, and 7 for the skeletal muscle. I'll be covering these two in 2 successive videos, first being the cardiac muscle and the next being the skeletal. Stay tuned and keep watching. Do like, share, subscribe my videos.